Hello, my name is Isabel Ramírez Acevedo. I am on third semester of medicine at the Pontifical Bolivariana University, and today I'm going to talk about iPSC-derived microglia as a model to study inflammation in idiopathic Parkinson's disease. So, what is Parkinson's disease? It is a neurodegenerative disease with unknown cause. That is why it is considered idiopathic. It affects dopaminergic neurons in the substantia nigra in the pars compacta. So, it is produced less dopamine, and it leads to shaking, stiffness, and difficulty with walking, balance, and coordination. It affects 50% more men than women, and advanced age is a main risk factor for the development. So in this study, they use iPSC-derived microglia. iPSC means induced pluripotent stem cells. It is a type of pluripotent stem cells derived from adult somatic cells, like blood cells or skin cells, and they reprogram them through inducing genes and other factors to make an embryonic pluripotent state that enables the development of an unlimited source of any human type of cell, like microglia. So the aim of this study was to investigate inflammation markers in different models of IPD, idiopathic Parkinson's disease. So different methods were used for this study. One of them is the quantitative PCR. It is a PCR-based technique to amplify a target DNA sequence with quantification of the concentration of the DNA species in the reaction. And in our study, they isolated RNA from microglia and they synthesized DNA. So they also use Western blotting that is used in research to separate a specific protein from a complex of proteins extracted from a cell. So they separate by size, then they transfer to a solid support, and they mark the target protein using antibodies. And in our study, they use the total proteins of microglia to isolate the inflammation markers. Another method was the immunocytochemistry that uses antibodies to check for certain antigens in a sample of cells. So these antibodies are usually linked to an enzyme or a dye. So after the antibodies binds to the antigen in the cell sample, this enzyme or dye is activated and the antigen can be seen under a microscope. And RNA sequencing is another method that provides an insight to the transcriptome of a cell. It is the collection of all the genes readouts present in a cell. And in our study, they use RNA sequencing to analyze postmortem midbrain tissues to assess the expression of different cytokines in IDP patients. So here are some results of the study. In figure A, we can see an overview of the protocol used to derive microglia from iPSCs. And in figure B, we can see some markers like NANONC, SOX2, and OCT4. And these are transcription factors essential to maintaining the pluripotent embryonic stem cell phenotype. And they use immunocytochemistry to characterize IPCS lines with the stem cells markers that I already mentioned, and we can see that in IPD the colors are brighter, so they achieve this embryonic stem cell phenotype. And also the differentiation of iPSCs into microglia was achieved with the addition of multiple factors through the differentiation process to mimic microglia development in the human embryo, and that we can see in figure A. Okay, here are more of the results. In the picture from above, we can see that they compared the expression of NLRP3 between the control cells and IPD. NLRP3 is an inflammasome. That means it is a complex of proteins that are sensors and mediators of the inflammation. IDP microglia were more reactive after priming with LPS. LPS means lipopolysaccharide, and it activates caspase 1 of the NLRP3 inflammasome. That is why there was a higher expression of mRNA and protein levels of NLRP3. But although the author said that IPD show a higher expression, we can see in the picture that the control cells is darker. Okay. In the other picture, they measure the expression of the LRRK2, that is lysine-rich repeat kinase 2, and we can see that IDP microglia have a significant decrease of this kinase expression compared to control cells, and LRRK2 may be crucially involved in the regulation of the neuroinflammatory process. That is why it's downregulated in IPD compared to control cells. Okay, now the discussion. McGeer et al. Said that inflammation has been considered a hallmark of PD since the latest 1980s, when an upregulation of reactive microglia was first seen in patient brain tissue samples. That agrees with our study. Simon Fick et al., Sharma et al., and Gielmaser et al. Also said that studies investigating LRRK2 expression in IPD brain tissue shows a significant downregulation in dopaminergic neurons, which may contribute to the pathology of the movement disorder. It actually agrees with the results of the study. Shunk et al., Gordon et al., G et al., and Pick et al. Said that higher levels of IL-1B and NLRP3 in IDP microglia indicate a stronger priming step, which is necessary from downstream inflammation activation and immune response, and it actually agrees with the results. Finally, here are my conclusions. We can say that molecular biology techniques helps to understand the pathophysiology of disease through the investigation of genes expression, DNA, RNA, and proteins, and that is the central dogma of molecular biology, and also the influence of environmental factors that can trigger a disease like IPD, and also with the investigation of cytokines that are involved in the development of a disease. Therapeutic options can be created to treat patients with genetic predispositions to develop one of these diseases. Thank you very much.